Okay, today I'll use this trinity, the science, engineering, technology trinity, as sort of a theme that we're going to develop through the whole term. And we'll just briefly refresh ourselves or remind ourselves what Professor Laufenberger talked about yesterday or on Tuesday. And then I'm going to go a little bit in some detail about how information is used in biology, in cells. It's a way of gaining sort of a broad perspective of a facet of the biological basis for bioengineering. But it's also a way to introduce to you how we can use that information in developing technology. Okay, now, just a few odds and ends. We do have a course website. On that course website will be articles that I may or may not download from newspapers or magazines. Generally, if you want to be in the biospace, read the newspapers. Today the concern is the occurrence of the flu virus in Africa now. So it's now spread from China to Turkey, from the Middle East down to Africa. There's a big question of how it got there, and then what's the implications for a flu pandemic. Of course, we have this very large concern about a flu pandemic, and as biologists, bioengineers, and biotechnologists, it's an area in which our contribution may be, in some small part, may play a large role. Okay, so just generally be aware of what is going on in the world. We have online, on the website, videos that the various bioengineering professors have produced that describes some facet of their research. These are professionally produced videos, so please go to them because they'll be useful in augmenting some of the seminars that you'll hear from the other faculty. I went over the paper. The paper will be graded, generally A, B, C, or fail. Last year, I think I gave an obscene percentage of A's. It's only a 5-page paper, so it's not like a writing requirement thing where you sit up the night before and write something. You really have to talk to me about what topic do you want to write about and how you are thinking about exploring that topic in your paper. So there'll be a certain amount of interchange between you and me about your paper, and as a result, you'll have a well-crafted paper that will be relevant to the kinds of things that we're trying to accomplish or learn in this course. This is just the schedule of the upcoming lectures, and so these are the professors from different departments in engineering that will be talking about some flavor of bioengineering. And course ends on March 23rd. That's when the paper will be due. It'll be due in class. I'd like also to have an email copy, but we'll go into that detail much later in the term. So this is only a half-term course, and I'll end with a selected topic in bioengineering, and it's usually something that's topical based on what's in the news. Okay, now Doug spent quite a lot of time emphasizing that engineers are both scientists and technologists. We create technology, but the technology is based on science, on knowledge. So science is what we can find out about something that exists. Engineers either learn from that and develop technology or help understand the basis for that science. What I'd like to do today is to explore the bioscience basis for bioengineering. And when you talk about biology, you really have to start with the cell. You can talk about the molecules that comprise the cell. You can talk about how cells are organized into tissues and organs. But in the end, the quantal unit of life is the cell. And so if you look at just the scale in which we immediately find ourselves in when we talk about cells, we're in the microscopic world. We're in the micron to tens of micron sizes, because that's typically what are the range and sizes of cells. And then if we think about how biology or biological structure is organized relative to cells, there's how tissues, which are how cells are organized to carry out a specific function. And then the tissues themselves are organized into organs. In higher animals, organs carry out a complexity of tasks. And obviously, the hierarchy goes to the body plan of an adult. Cells, on the other hand, are themselves, we'll view them as machines that are composed of subsystems. The subsystems are built from basic components. The basic components themselves are some specified collection of atoms. So obviously, we can go down to the nanoscale. But when we talk about nanoscale in terms of biology, it's really at the level of the macromolecules that comprise biological structure. So the size range is huge when you talk about biology. It's from nanometers to millimeters, right? To meters, sorry, in size. We can think about biology in terms of energy that drives or that is the basis for biological function. We can talk about the forces that are involved in biology, whether it's the forces that are exerted at the cellular level or forces that are exerted at the molecular level. So as opposed to the physical world, the biological world is very much a world in the piconewton to nanonewton range. So it's not a lot of force. Piconewton is roughly the force exerted on a bacterium by gravity. Or if I had a laser pointer and you saw the spot on the wall, it would be the optical pressure of the flux of photons on the wall. That's on the order of a piconewton. So we're not talking about a lot of force, but at the molecular level, that's what drives biology. As far as times, biology is very much a chemistry-based process, and so chemical reactions and things that regulate and control chemistry in those time scales. But it's also a mechanical process. And so there are mechanical steps and mechanical reactions at the molecular and cellular level that have their own time domain. And then obviously there's other time dimensions. For instance, the onset of cancer is measured in years as opposed to things that happen at the molecular level. Okay, so just be aware of the space, that we'll be talking about in different examples. I have this picture that I lifted from David Goodsell's book. It's a rendering of a bacterial cell in which he has depicted the components or the contents of that cell in as realistic fashion as possible. So it's the dimensions or the sizes of the macromolecules, in this case the protein and the nucleic acid is represented, as well as the concentration of these elements. And so one of the striking things that you see is that a cell is packed or is a dense collection of macromolecules. To your eye, it may seem that this order is random, but in fact there is order in the organization of the cell and its contents. As I said, cells range in size. They, in a lot of cases, move at different rates. Their lifetimes, depending on what cells, can span from minutes to years. We already talked about force. Living systems basically combat entropy, so that requires energy of some kind. The energy is converted from energy sources that we ingest or we absorb from the environment. There's some basic activities that cells do. All cells grow, divide, and die, right? And then, in addition to that, they take on some advanced functions or specialized functions, like an immune cell will migrate and look for pathogens in an organism. A muscle cell will contract. A nerve cell will transmit information as electrical pulses, and so on. Just to give you an idea of the magnitude of biological complexity, a bacterial cell is a unicellular organism, so its unit is one. whereas an estimate for the number of cells in a human is on the order of 10 to the 13th. So that's you and me. And in our lifetime, we turn over on the order of about 10 to the 16th cells. So that's a lot of biology that goes on in the span in a human lifetime. Now, we're engineers. Okay, you're here because you're interested in bioengineering, and there's an aspect to understanding biological function at the cellular level, at the molecular level, or on the other hand, at the tissue or organ level. It'll be emphasized to you over and over again that you should view these things as systems, please. Actually, everybody turn off your cell phones, too. Okay, so if we think about cellular systems in biology, we can think of it in terms of the different kinds of cells that may make up an organism or an animal. And one way of thinking about it is to break down biological processes in sort of basic units. And we tend to think of them in terms of the chemical systems of a cell, mechanical systems of a cell. So the chemical systems are basically the biochemistry of cellular processes. The mechanics describe all of the forces and structures that contribute to force that produce motion, either at the molecular level or at the cellular level or at higher levels. We know, for instance, that cell membranes have the ability to segregate charge. When you distribute charge, then you set up an electrical system. And then we're living beings. We, as I said, try to combat entropy. And so there's a certain amount of thermal energy that characterizes living systems that is the input from the environment. And actually that amount of thermal energy from the environment accounts for a lot of the stochastic or random behavior that you see in biological processes. So, as a formalism, we can think of cellular systems or cellular machines as doing work. And then the question is, whether or not we can describe a cell in terms of its subsystems and whether that's a good representation. So when Doug talked about models as an endpoint of the synthesis between experimental biology and engineering, one of the things that we want to do is to understand these different subsystems at to a degree that maybe we are able to, in the end, model a cell, either at a very basic level or at a very fine level. Because if we can model cellular cells or cellular processes, then if you think ahead, we can then devise or think about ways in which we can interfere or intervene or modify the behavior of cells or what cells do or things that go on in cells. Okay. Now, I told you a couple minutes ago that cells grow and they divide, and after they divide, they become something. Okay? And in a human being, you start with a fertilized egg, which grows and divides into 10 to the 13th cells. So in the development of an embryo, there's a lot of cell divisions that go on. And in the lifetime of a person, if there's 10 to the 16th cells, then that means that there's on the order of 10 to the 16th cell divisions that go on. So 10 to the 16 is a pretty big number. And at the end of the day, or at the end of the year, or at the end of the decade, or at the end of some point in your lifetime, you pretty much want to make sure or hope that the cells that are being replicated in your body as you age actually have exhibit a high degree of fidelity in copying the initial set of information that gave rise to the cells to your cells when you were an embryo. Because if there are errors in the information that specified the division of cells into two daughter cells, then those errors get replicated. and additional errors are introduced, they multiply. And there may be synergy between these errors, which we call mutations, that lead to, for instance, cancer. So if you think of this as, in terms of information, the cell is somewhat of an autonomous system, and the cell grows, has the information for dividing into two cells, and it has the capacity of copying that information that specifies a cell into the two copies. Okay? So this is a little bit different way of thinking about what we call DNA replication and cell division than how you may have heard about it, for instance, in 701. So that raises a question. How many have taken 701 or are taking it now? So let's put it this way. Who hasn't had introductory biology? And then who hasn't had a biology course? Okay, that's all right. Okay, so in 10 to the 13th mitoses to give rise to the adult body plan, or the 10 to the 16th divisions that give rise to cells in your entire lifetime, you want to make sure that the information that encodes this process is copied faithfully without error. Now, we know what that information is. It's your genome. It's the organization of DNA, the sequence of DNA in the cell. And the genome is, the DNA encodes, or you can think of DNA as a chemical tape, as an analog of a type of a tape. But in this case, it's of a chemical nature in which the sequence of just 4 bases specifies what ends up being the components list of the cell. So the genome, tells us what different proteins are encoded in the cell. Also in the genome are sequences that tell us where these protein or genes that encode these proteins lie, and some aspect of the timing in which these genes are read out. And then the other thing about the genome is that it's actually mirrored, right? It's not a single strand of DNA, but it's a double strand of DNA. And so if you remember back to even high school biology, that double strand is a type of mirroring because one strand is a complement of the other, meaning that if you only have one strand, you can synthesize the information or the sequence on the other strand. So you can think of the genome itself, because it's a diploid copy, at least in us, as a way of providing some error checking capability. But also then in each cell, you inherit almost a 100% accurate copy of your genome. There is an error rate in DNA replication, and we'll talk about that in a minute. Okay, so there's cells grow and divide. Some cells actually stay as a resident population of stem cells, which just grow and divide, grow and divide, grow and divide, spewing out cells which later will become specialized into different kinds of cells. And so here is a representation of cells that give rise to the cells in our immune system and our blood system. So here's the T and B cells from lymphoid stem cells, which originate from these pluripotent stem cells. And then red blood cells or platelets or other cells that arise again from this pluripotent stem cell, but through a myeloid stem cell lineage. So there are decisions that are made whether or not to stay or remain a stem cell. And then if the decision is to become a differentiated cell, there's decisions that control, or steps that control whether a cell ultimately becomes one of these kinds of cells or becomes cells of the immune system, and so on. So that's a general plan of how complexity is carried out in these systems. Now, I talked about the DNA is how information is represented, and it's packaged into genomes. The genomes themselves can be one piece of DNA, or for very large genomes, it's convenient to break them into smaller bits called chromosomes. This is just a plot of the different genomes that actually have been sequenced over the last 25 years or 30 years, in which the size of the genome is represented in the number of nucleotides in that genome. Or another way of thinking about how big a genome is in its functional units, which are the genes. And so the human genome is 3 billion bases. It encodes roughly 24,000 genes. If I gave this, if you sat in on this lecture maybe 5 years ago, certainly 10 years ago, we had thought that the genome was more like 100,000 genes. But now that the genome has been sequenced, it's now certain that the number is much smaller and very close to 24,000 genes. These blue dots represent key genomes that have been sequenced with time. And so you see that we started with a simple virus, went to a simple organelle, went to more complex viruses, and then eventually up the tree of complexity to yeast, and then metazoans such as flies, worms, and humans. In the meantime, genomes have been continued to be sequenced, and the community of genome sequencing people have spent a lot of time sequencing microorganisms, since they're easy to sequence and the biological diversity is high, and plus because we're interested in combating disease. Okay, now, again, going back to what you should have learned already, this is just a schematic representation of DNA. This is a diagram of the chemical structures of DNA, in which the 4 different bases are represented by different colors. And what this diagram represents or shows is that what one strand encodes is mirrored in the other, because one particular base will base pair with only one of the other 3 bases. So we have, in double-stranded DNA, an exact complement of each strand. And then, again, if you think of this in terms of a tape, when we want to copy the genome, it's a physical process. So think of it as a tape machine in which you copy from one tape to another. There's a readout or a reader, which in this case is an enzyme complex called DNA polymerase, and other cofactors and other associated proteins. That enzyme or that machine will take DNA. DNA has to spool through the polymerase, but this is a chemical process too. So you need nucleotides to be fed in. They get assembled in some covalent, through covalent bonds, and you get from one strand of DNA its complement, and from the other strand, that strand's complement is also synthesized. So think of this as a machine. The machine does chemistry. The chemistry synthesis is a chemistry in which a chemical copy of a chemical tape is made, but that there has to be a process in which this machine reads the information or recognizes each base at each position and inserts with high fidelity its complement. Okay, now the error rate in in chemical synthesis is on the order of 1 in 10 or 1 in 100. Okay, so if you think of making DNA with a machine or just taking a simple set of chemicals and throwing them together and throwing in an enzyme that copies them, you get a pretty good copy of DNA. 90% accurate is pretty good, right? But when you think of your genome as being 3 billion bases, 10% error means that you have 100 or 300 million. You have 300 million. Sorry, 10% error means that 300 million bases are in fact wrong. So obviously we can't tolerate that rate or that level of error. So the genome is read by polymerase. It reads or synthesizes a complementary strand at the rate of about 800 or 1,000 nucleotides per second. For very large genomes, then, that would take a long time. In our body, a human cell divides approximately once a day. And so we have 3 billion bases. You could do the math. Your cell, you don't have a lot of time to do a lot of things. So for very large genomes, the replication proceeds in parallel at different places within a chromosome, and all the chromosomes are being replicated simultaneously. So you have this process in which you make a copy of the genome, and then those copies are then distributed to the daughter cells during cell division. Now, as I said, the frequency and error in just making a chemical copy is on the order of 1 in 100 or 1 in 10. There's additional sources in the process that improves this error rate. So, of course, we can't live. We wouldn't be able to survive if our DNA was mutated at that high rate. And so, for instance, as a chemical machine that adds a nucleotide that's designed to base pair with an existing nucleotide in a DNA strand, the polymerase has a selectivity for certain nucleotides given what base it needs to insert. And so that selectivity reduces the error rate so that at the level of polymerase, the error rate becomes 1 in 10,000 or 1 in 100,000. That's still pretty high. So what do we have, or what's available to improve the fidelity in DNA replication? The polymerase, or one of the associated proteins, actually checks which base got stuck in. And it looks to see what base it should have stuck in from reading the original strand, and it sees what base actually got stuck in. And guess what it does? The wrong base got stuck in, it stops, takes that base out, and replaces it with the correct base. So there's a proofreading function, and that improves the fidelity of replication by several log orders of magnitude more. So we're moving in the right direction, right? So that's problems in just the inherent machinery that we have in copying DNA from one strand to another. Now, on top of that, there are a whole load of other factors which are trying to mutate that DNA. And a lot of the problems or a lot of the causes of cancer are environmental influences that cause mutations in our genome. So, for instance, I fly a lot of miles every year. If you're in a jet at 40,000 feet, my exposure to ionizing radiation is a little bit more than yours. Or if you spend a lot of time near sources of radiation, then your DNA is going to get damaged. There's mechanisms that look for damaged DNA where there's a chemical change in the DNA base in your genome, or there could be places in which a base got deleted or inserted. Anyway, there's, in addition to the error correction that's inherent in the replication machinery, there's post-replication error correction processes, one called mismatch repair, and that goes back and further cleans up the genome sequence. And so the frequency of error after mismatch repair is now down to a level of 1 in a billion or 1 in 10 billion. So, you know, the evolution of the system in faithfully copying one genome into 2 copies is actually a very finely honed process, and it's all through evolution. Now, if you think of this, I don't think there's a physical system. If you copy from tape or copy onto disk, you don't achieve this high level of fidelity in those copies. Okay, now, all I told you about was that information got copied from one strand or one genome to another, and that the information is in genes. But that in itself doesn't tell you anything about the machinery. All we know from the genome is the parts list. All we know is that there's 24,000 genes. We have a good idea of the function of many of those genes. We don't know what all of those genes are or what they do. We have a good guess, and we are continuing to be able to make better guesses about what those unknown genes are up to. But in the end, the genes encode proteins, and the proteins are the execution machinery of a cell. Those are the macromolecules that actually carry out the work of the cell. And so the question then is, how do you go from information encoded in DNA to somehow information which is really just function? All you care about is that different proteins carry out specific functions under certain conditions. How do you go from a string of 4 bases to a function? And so, again, in biology, you've learned through the process of transcription, in which a messenger RNA is made, and then the process of translation, in which the information in nucleic acid sequence is then translated into a protein sequence, that eventually protein function arises. So it's an odd concept, right? It's like taking a blueprint, the parts list of of a machine, and somehow trying to figure out how that parts list knows what to do. Okay, now there's an important concept that you have to keep in mind when you're trying to study or understand biological process. In that we have several levels of interactions that are responsible for encoding the information. First of all, in the case of DNA sequence, it's the string of bases and the sequence of that string of bases which is important. But that sequence is hardwired in that each base is covalently attached to its neighbor. So it's a chemical bond which holds the information in its place. Now, what happens is that that information is then translated into a protein sequence, a sequence of amino acids, and they're collinear in their information. So the protein sequence, if this represents a polypeptide chain, is a string of amino acids also held together by covalent bonds. But that in itself doesn't encode or doesn't represent a function that's carried out. What happens is that the amino acids in the polypeptide chain, at least in a local area, will tend to interact with each other in specified ways that we understand very well. And what you have is local occurrences of the polypeptide chain aggregating or assembling or associating with each other in very stereotypic ways. And so you have local organization of polypeptide, and then these elements then further associate with each other and compact into something that has ultimately a three-dimensional structure. So you have the information originally hardwired in a sequence, but the nature of the amino acids that are in that sequence then causes that protein to fold. Once that protein folds, it's only when the protein is folded does it become functional. So that's how information encoded in a genome ultimately encodes or inherently has in it the information what to do. Okay. Now, this is a diagram of how genes are turned on and off. And it's a particular network diagram. When a phage infects a bacterial cell, there's one of two choices that's made. The phage either decides to integrate itself in the bacterial genome and hibernate, or if the environmental conditions, i.e. the nutrients, are high, then that phage will replicate and generate new phage. And so this is the decision circuitry for that, in which we have, in blue, the DNA sequence encoding, in this case, the genes for different elements, different proteins. In front of these genes are sequences in which other proteins bind, and those proteins, whether they're bound in front of a gene or not, determines whether that gene is turned on or is inactive. And you see from these arrows that genes that get are activate when genes are expressed and protein made, that those genes, in combination with other genes, can form a decision local logic circuit, which then tells other genes to either turn on or turn off. Okay, so you can look at how genes interact and understand and codify in very specific ways the biochemistry of a particular process. And so this is a model. It's a representation of how we see, in this case, gene expression is controlled. And because it's a model, we can then predict or simulate what the outputs would be, whether it's a prediction of whether the bacterial cell or the virus will replicate or whether it'll hibernate. Okay? So this is, again, a role in which bioengineering plays, because it is the element in which we can not only analyze biological processes, but one of the goals, if you remember, in the design and synthesis aspects of what engineers do, modeling is an important component in design. Okay, now, in the last 10 minutes or so, let's now take this little bit of information. I walked you through, in a very gross level, how DNA is replicated. It's through polymerase. Now, let's see what we can do with that little finding or that little bit of information and turn it into something that has a technological application, which is DNA fingerprinting. So how many watch, what's that show? CSI. I think more people watch CSI. Just don't be afraid. Just raise your hand. We know you're all closet TV freaks. So, okay. Well, anyway, just think about DNA fingerprinting or genotyping as an application. The background is this is a chromosome, has 2 arms, and just on chromosome 11 in our genomes, there's a location called TH01. Okay? It's a very specific location in the DNA sequence. Okay? Now, first of all, I want everybody to think of 2 numbers between 6 and 12. Okay? So the first number we'll call M. And so everybody think of a number between 6 and 12, write it down. M and then the number. And then everybody now think of a second number, we'll call it F. And again, pick a number at random between 6 and 12. This is how, actually 5 and 11. Sorry, we'll do 5 and 11. We'll do TH01. Okay. Okay, so this is how DNA fingerprinting works. And it's based on the discovery that in our genome, we have areas in which, in this case, a 4-base sequence, AATG, happens to be replicated, happens to be copied in tandem. And at this position in all of our chromosomes, there'll be anywhere between 5 and 11 copies. Now, we're all diploids, right? We got one copy from mom, and we got one copy from dad. So on one chromosome inherited from mom, there's some number of repeats. And on the other copy inherited from dad, there's another set of repeats. So the number of repeats of these sequences determines which allele at that locus that you have. Now, these repeats in our sequences aren't just confined to this one part in chromosome 11, but in fact, there's millions of these kinds of repeats. They range in size from 2 base repeats to 6, 8 base repeats. So this is just a map of the distribution of some commonly used 4 base repeats that are used in diagnostics and in determining whether you or someone are related or are responsible for the origin or source of a particular piece of DNA. So in all of our chromosomes, we all have these simple tandem repeats. And so the question then in distinguishing, just say you from somebody else. So here's the scenario. There was a crime, and there's a pool of blood. We take, just say somebody smashed some glass, grabbed something, got cut, left some blood on the glass. We analyze the DNA. Okay. Now, what we analyze the DNA for is the alleles at TH01. So how many people picked, I'll say 7 as one of the alleles? Raise your hand. My goodness, that's more than statistic, well. Okay, then how many picked 10 as the other allele? Okay, not as many. Now, how many picked both 7 and 10 as both alleles? So that's even less. Okay, we have 1, 2, 3, 4, 5, 6, 7. Okay, now, obviously, at random chance, at one locus, you matched at those 2 alleles, right? Now, does that mean that you committed that crime? And then think of the world population. How many billion is the world population? At random chance, if we were only to look at one allele, how many people would match just if we assayed at that one site? Okay, so what's the answer? Obviously, if we look at other sites in our genome for other repeats, then we can knock down the number of incidences in which you, by random chance, would match with that sample. But even with the piece of DNA that, if just looking at that loci, we can still knock down the number of people that match. So how many people, so 711, what was it, 710, right? 710, let's just say M is 7 and P is 10. Now how many match at those 2 loci? Not just 7 and 10, but that the 7 was matched with the M and just 1? Raise your hands high. 1, 2, 3, 4, 5. Okay, so we knocked out 2 people, right? So if we went to your mom and we went to your dad, got their DNA samples, then you should have, well, let's say your DNA should have at least one of the alleles contributed by mom or dad. Now, there would be a problem, right? If if one of the alleles didn't match mom or dad, it means that either mom or dad wasn't your biological mother or father, and that happens. If you if you watch on public TV, there's a series by William Lewis Gates, a professor at Harvard, who genotyped the DNA of 8 prominent Afro-Americans, including Oprah Winfrey, and then revealed to them where their biological origins were. So it's all about DNA testing. Okay, so here the name of the game is to determine how many repeats there are in your genome, and so it's just a counting exercise. And so now the technology is, how do we count repeats? How do we find? We know where to find the repeats because we can use polymerase to copy using a strand that's designed to flank the regions that outlie these satellites of repeats. And we use polymerase to make a copy of one strand and the other. So this is the polymerase chain reaction. And the number of repeats is directly correlated with the length of that copy, the replimer. Okay? And so if we take a piece of DNA that has these repeats, throw in these primers, amplify across this region, we should be able to see in that DNA sample, we should be able to, based on size, tell whether or not it correlates with one of the repeats in a model DNA sample. So this is just a sizing ladder of 5, 6, 7, 8, 9, 10, 11 repeats. And these are samples taken from different people in which this corresponds to 6 repeats. This corresponds to 9 repeats. This corresponds to 8 repeats. This corresponds to something a little bit less than 10. It turns out at TH01, you're missing one base, so that repeat turns out to be a little bit shorter. So in almost all of these cases, the alleles are different. You got one from mom and one from dad. They turn out to be different. In some cases, they happen to be the same, so you get one band. Okay, so it's counting these repeats. Now, of course, we want to measure at different loci to exclude people at random. And so we want to look at different loci. We also want to increase the efficiency of this assay, so we multiplex the assay. Instead of looking at just one repeat, we look at 5 or 6 repeats all in the same assay. And so what we do is we size, separate the different repeats of the different loci on a gel. So it's a medium for separating DNA by size. And so you see here the alleles for penta E, the alleles for this locus, the alleles for TH01. So just running on one gel, you can discriminate among 5 different loci. If you run 2 gels, you can discriminate among 9 loci. And the standard test done in DNA forensics is looking at something like 12 loci. So at 12 loci, the chances that you will match with a sample left at a crime scene is scales on the order of the number of loci that you test at. So if you test at 12 loci, then if you have a world population of 10 to the 12th individuals, then there's a random chance that 2 people will match in their DNA. So this is virtually a foolproof method for identifying identity. I'm going to speed through the rest of it. If it's a matter of sizing by DNA, then we understand how that process occurs. It's a matter of how long the gel is. It's a matter of what the selectivity of the gel is for separating one piece of DNA from another. It's a matter of how you inject the DNA in, and there's an element of diffusion. So let's hold the thought. We'll go back to this and quickly finish it up on Tuesday, and then I'll introduce you to the next topic.